action.
All right, so this is all informal. I was told to start by reminding everybody to sign up for the office hours as well. I'm going to talk about historical information in clinical trials. So using old clinical trial data or really anything else in the context of doing a design and getting more efficiency. There is a paper on this that appeared in Pharmaceutical Statistics that has 400,000 authors on it. The authors do include FDA, lots of industry, academics, includes people from SAS. Anyway, it's broad consensus. This is explicitly accepted in the device guidance at FDA, and it has been publicly accepted by senior statistical leadership in terms of acceptability as well. Now that said, there's context specific. Knowing you can do a design doesn't mean it's appropriate in every case. So I want to talk a little bit about the pros and cons of doing this. Whenever you do a trial, I'm going to just talk about the context of comparing a novel treatment to a control arm, pretty standard setting. This does generalize. The control is rarely existing in a vacuum. We typically always have data. If we novel treatment, we've got a control arm. At some point in the past, that control arm was the novel treatment. There are those kind of clinical trials. There are other trials that may be going on right now. So drug B may be trying to beat the control arm, but last year drug A tried to beat the control arm and failed, for example. Those kind of trials are out there as well. There's also registries, comparative effectiveness studies. All of this may be sitting around. This is a big deal in rare diseases, where a lot of times there's been a patient registry in a rare disease, and they've got longitudinal data. Here's some notion of what the course of the disease is. And if you're going to enroll a trial, you really want to leverage every last bit of information, especially in a rare disease setting. But this isn't specific to rare diseases. So prior to the study, we often do have a good idea of what the control arm is going to look like. We think it's going to look like whatever the data was before, but of course we also recognize we're running a new study. We have specific inclusion and exclusion criteria. The sites are a little bit different. It's the same, but it's not the same in certain ways. So how do we use that information? I wanted a couple seconds on nomenclature. You can get really visceral results from people when you say historical whatever. If you're talking, I'm talking mostly about historical borrowing here, which is a notion that refers to I'm running a randomized trial and I'm trying to augment the information in the control arm with history. There's a difference between doing that and what you'll often hear on historical control, which typically refers to a single arm trial, which you're basically going, hey, in the past, this is what the control arm acted like. I'm going to pick the number from that prior trial and try to beat that number. You can think of historical control, the single arm, as a special case of historical borrowing, where you really are hyper-weighting the historical data so much that you don't care what you're doing now. But this is a far riskier setting to just do a single arm trial, and I'll show why, than doing the historical borrowing. So a lot of times when I talk to people and I say we're going to talk about doing historical borrowing, their first reaction is, oh, you can't possibly do that because I need controls, or vice versa. So just keep in mind that terminology is out there. So which studies can you borrow information from? This is actually huge. I'm not going to spend a lot of time on this, but you have to have picked the right studies. To some extent, you don't want to pick, I'm going to do a study on people who have a certain kind of comorbidity, and I'm going to borrow from a study that was done in Europe that was on all comers, for example, because the specific population you're dealing with that has the comorbidity, they could actually be qualitatively different. So you do have to worry about that. You have to worry if you look for a literature search, are they actually representative? You need to worry, are they cherry-picked? So you don't want to get your I'm going to pick, here are the 2 studies that worked, and I'm not picking the 7 studies that didn't. This is a lot of the same kind of criteria that comes up in doing a good meta-analysis, for example. This is, and in fact, a lot of our methods look like meta-analyses to some extent, so this matters. They also shouldn't be cherry-picked, and this comes up in weird ways. Had a client, I guess now a year ago, but anyway, they came in and they said, we want to do historical borrowing in this population, and we've done a prior study in this population, and here are 50 subjects of data. So now, can we use those going forward? This was a fairly rare disease. We want to leverage this. We don't want to reinvent the wheel. All makes sense. Their studies seem to be high quality. It was on point, but I ask questions, a few questions, try to figure out what's really going on. And about a half an hour later, this wasn't the only 50 patients we had. In fact, we did a 300-patient study, and we did about 17 subset analyses, and this is the subset of patients that did the best. And so we're showing you the 50 patients in the subset that did the best. Well, that's a big deal, because if you do the subset analyses, you clearly created an upward bias in what you selected. And as we'll show as we get through the details, it's actually that upward bias is in a direction that causes some angst, especially from the regulatory perspective. So you want to understand really where this data came from and why are you seeing this historical data and you're not seeing that historical data. So this is one reason you could see, if I realize we're mostly academic here, but from a regulatory perspective, this is part of the pushback. It's not methodological. It's, I don't quite trust the historical data that you're actually using in these cases. A lot of times people will look through patient records at sites prior to study commencement. This, of course, has its own set of possible biases. One situation I really like, that we've been doing a lot lately, is platform trials. I think this is very promising for this, especially historical data. In a platform trial, you're talking about something that's been continuously running. So a group of sites they've got patient streams coming in, and multiple treatments have been coming in over time. So we've got a trial called I-SPY 2. It's in breast cancer, and that's been running for not quite 10 years, but something on that pace. And at this point, what we've seen is we've got 10 years of historical data, and it's not in a separate trial. It's actually at this trial. They're at these sites. They have the patient population's been pretty stable over time. And in addition, we can statistically test this. So we can plot this trend of historical data and see that it's been stable over time. There haven't been jumps. Sometimes that's disrupted. The control arm in I-SPY 2 just changed because the standard of care changed because of pertuzumab. So anyway, that kind of stuff, you can get disruptions in this, but that kind of historical data is good because there's an explicit method for testing its applicability, and we model that control arm all the way over time in order to use it, and it's quite efficient. So that's been one of the more promising ways of avoiding biases. All right, so simple example. I'm going to talk about a dichotomous endpoint. We've got 200 subjects on each of the control and treatment arms. So just binomial data, yes, no for everybody. I've got 2 rates. So I've got a rate in the control arm, that's PC. I've got a rate in the treatment arm, PT. I've also got historical data, so some trial, I'm going to assume that it was well done and applicable, and the only one that I have, I'm not omitting, so assume this has been done well. But I only found one historical data set, and it had 65 out of 100 responses, and this is on the control arm. We're talking about borrowing from the control. You can have a separate discussion on borrowing on treatment. That's more controversial, but you can, in theory, do it. So here I've got the H's for the historical data. When you see it, there was some rate in the historical study which I'm calling pH. So I actually have 3 rates here. I've got my rate for the control in the current study, and I've got pH, which was my rate for the control in the historical study. I'm hoping those were close because they were done under similar situation, but they're not guaranteed to be. And then PT is my treatment rate. I'm doing a standard kind of test, so within my current control arm, I want to test is PC equal versus PC being less, so I want my rates to go up. The basic idea here is we're going to combine information between the current control study and the historical study. So I'm going to have 200 current subjects, and I also have 100 historical subjects. So I want to augment my control arm with those 100 historical subjects. So when you do this, this can be good and can be bad. And the problem is that you don't know in advance which one it's going to be. So you're taking a risk in doing this, and your hope is that the odds you're going to get the benefits exceed the chance that you're going to pay the price. If the historical information is close to the true parameters, then your inferences are improved in this case. And by inferences improved, I mean you get higher power, you actually get lower type 1 error, and you get smaller mean square error. So in terms of all the things we often care about as statisticians, everything's better in this case. It does behave in a better sense. On the other hand, if the historical information is far from the true parameters, then you can get biases and other kinds of problems. If it's off in either direction, you get biases. If it's off in one direction, you can actually reduce the power of your study by doing this. And if it's off in the other direction, you can increase the type 1 error. I'll show some graphs to this effect in a moment. So again, the issue is we don't know in advance how much drift we actually have. So you can think of kind of the checklist on what to pay attention to. I've got this true current parameter, so this is that PC. In the middle, I've got the true historical parameter, that's PH, whatever it was. And then I've also got the historical data, so that's YH, there's a difference here. The key thing that makes a difference is the difference all the way across. So that drives the operating characteristic, the difference between the observed data and the true current parameter. I like putting this in the middle because it's good to kind of get an idea of why these two things are different. This piece of it is almost entirely sampling variability, assuming that you've done things right. So this is something we understand a lot about as statisticians. This other piece is differences between the true parameters, which gets a lot more into substantive clinical discussions. So the standard of care might be changing over time. The other study was done 12 years ago, and for whatever reason, people are just doing better today. That would be a form of drift. You could have the standard of care could qualitatively change. So it used to be they were doing X, but now it's X is always combined with Y, and patients are doing better. You've got the usual stuff, different sites, different patient populations, all of the stuff that just, hey, studies are going to be a little bit different, and these are common to meta-analysis settings as well. And again, be careful about this cherry-picking kind of issue, because that can always create systematic issue biases really kind of all over the place. The simplest way this gets done is through what's typically called downweighting. And this is a form where every historical subject counts and you assign a weight to it. And that weight can vary depending on whether you want to heavily consider the historical data or whether you want to minimally do it. So if you give it a weight of 0, you're essentially ignoring the historical data. If you give it a weight of 1, that's like 100%, that's going to result in an analysis that's similar to pooling. So you'll end up basically counting everybody. A control and historical subject are all the same. Usually weights less than 1 are picked here, so that's called downweighting. So we might say we're going to count each of the historical subjects as 1/2 or 1/4 or 1/5 of a current patient. And this doesn't actually get done very often. In theory, you could overweight them. I'm going to count the the historical subjects as twice as valuable as the current ones. It's kind of bizarre. But I am emphasizing this because if you do all the math, a single arm trial actually corresponds to a weight of infinity. So people do overweight all the time. They just do it in this context of, okay, 0 is okay and infinity is okay, but now I'm nervous about everything in the middle.
Is there a downweight that's used more often?
Yeah, so I don't know that, there's no downweight that I would consider has any completely scientific rationale. Actually, it's I have seen analyses that have talked about weights in the 1/4 to 1/2 range being more valuable. Certainly they're used more. I don't know that I would consider any line in the sand that goes, this is right and that is wrong.
Yeah.
So question, if you're beginning your design with say power prior and you have chosen some way to weigh your priors, and when it times for inference, your effect is in the other direction. Would you still work with the non-informative priors to make the final inference, or would you go back to the prior you used for your design?
That's an excellent question. So one issue, you pick a weight, you can get to the end and decide this weight doesn't make a lot of sense now that I'm looking at the discrepancy between them. That can go in reverse. You could have picked a small weight and recognized, boy, you know, I got 65 out of 100 in the historical study, that's 65%. I'm getting 66% in my current study. Maybe I should have just, hey, everything's the same. Why didn't I just pull? So usually I actually don't use power priors.
Okay.
And I actually put a prior on the weight itself and generate a hierarchical model. So I'll get to that next. If you put in your protocol that you're going to use a power prior, I mean, you've got in your protocol, this is what your analysis method is. So you're stuck with it. I mean, you can do analyses later, but they have to have a label on them going, hey, I decided I didn't like my analysis plan after seeing the data, and that, that's a pretty big accuracy. So here's an example of this. Power priors, the way these are often done, this is the likelihood function, so this is the mathematical details. We just raise the likelihood function to a weight, and in effect this does the intuitive thing. So if W is equal to 0.2, for example, a weight of 1/5, what we effectively do is take that 65 out of 100, we're weighting 1/5, and we say, okay, that's 13 out of 20. And so in effect, what we do is we pull the equivalent of 13 out of 20, lift them up, put them in our current data set, and then we go on and do inferences as usual. So the math comes out in the wash. This applies to dichotomous data, continuous data. You can do similar things in time to event where it doesn't fall out quite as nicely. But essentially what you're doing is we often talk about the number of subjects that you are borrowing, the effective weight. And so the weight here would be 100 W. So here I'm borrowing the equivalent of 20 patients by picking a weight of 0.2. The inferences that you get, as you can imagine, your posterior means, for example, are a weighted average of depending on what your weight is. So if you've got 146 out of 200, so you got 73% responses on the control arm. Of course, if you don't borrow, your posterior mean 73%. If you have a weight of 0.2, you act as if not 146 out of 200, but 159 out of 220. And so that gets you 72%, and so on. And so you can see the weights gradually pulling this back towards the historical data. So we've got our 73% observed, the historical data will pull that back towards 65, and your weight's really telling you how far you're going to do it. If you overweight, you get back to about 65%. The more you weight this, you basically are ignoring your current controls, which is why a single arm trial is essentially a weight of infinity. If you had enrolled controls, you would have ignored them anyway. Operating characteristics of this trial. So we usually obtain the posterior distribution of PC, the current control rate. If we want to find the mean square error of such a thing, we can compute that. So just take that weighted average and we compute the mean square error through standard methods. We can also do hypothesis testing. So I'm not doing anything complicated here. I am being a Bayesian about it. But this is just rejecting if the posterior probability is sufficiently high. We can compute the type 1 error of that test, and we can also compute the power. So keep in mind, I've never quite figured out whether things are Bayesian or frequentist. As long as you compute the operating characteristics, everything's frequentist.
No one has.
So,
Right?
so anyway, I consider, when they kind of go, I'm like, don't you guys know that all the variance frequentists, that's all we do, because we have to the operating characteristics of the idea. So anyway. All right, so power and type 1 error, one key thing is they're a function of the current control rate. So we're used to test where they have, say, 2.5% type 1 error, and that's pretty invariable to what the control rate was because they scale. Here you've got this weighting system. So if you've got historical data at 65%, if my true parameter is over here, I'm pulling it up. Well, that changes the operating characteristics on that side because I'm always pulling the control arm up, and I'm making it harder to win. You pull the control arm up, the treatment arm has to work harder. So I make it harder to win the trial. On this side, if the controls, well, I'm pulling controls that would naturally be higher than 65%, I'm pulling them down. That makes it easier to win. So imagine, the treatment's got to jump over a bar. I pull the bar down, it's easier to jump over it. So you get the operating characteristics reflect that. So this is what the mean square error looks like for a number of different weights. So I've got a weight of 0 in here. So this is, actually I should say what the axes are. This axis, which I know you can't see the label, this goes from 50 to 80%. This is the true current control arm. So am I really enrolling a current population that has a 50% rate on controls, or 55 or 60 or 65 up to 80%? My actual historical data is right in the middle here, but this axis is what the true control is right now. And then the y-axis is the mean square error. So the orangish line, which doesn't quite look orange on this projector, but that's okay, is no borrowing. So that's your standard mean square error, and it's not quite flat because, of course, we know mean square error is maximized around 0.5 for a dichotomous variable. The pooled analysis, where I just take all the historical subjects and I count them full weight, W equal to 1, that's in red. So that's this analysis right here. And then I've also got the single arm trial that's in purple. And then the other weights that are in blue, they correspond to, how many do I have? I got to figure out. So I think it's 0.2, 0.4, 0.6, and 0.8. So you can see the continuum here. And these certainly, if you start with a weight of 0, this does form a continuum. You gradually start from this kind of thing and you push down to that as you increase the weight. You can see there's a couple regions here. So there's a region right in the middle. If you don't have any drift, your true control proportion matches the 65% in history, you get gains in mean square error. So for example, the pooled analysis can cut the mean square error in half if you're right on target. The single arm trial, if you haven't had any drift, you should absolutely do a single arm trial. It cuts the mean square error down to a fraction of what it was. And this is on the control arm. The difficulty with this, of course, is as you move away, you generate biases. You're pulling things away from what they actually are. You've got essentially a bias variance trade-off. You're always going to introduce bias. It might be small, might be large, but you always save variance because you are adding something into the study. So you get kind of a sweet spot here, and how big the sweet spot is depends on the weight. So, this low weight of 20%, it doesn't give you a lot of benefit, but it gets you a benefit over a very broad range. So there's some value here, unless you're off by a lot. I often find in analyzing, I do this a lot in early phase 2 studies where they've got a relatively small sample size. The variance gain from using the historical data is typically pretty large. And the bias issues, because the variance in a small study is so large and getting rid of that variance is so valuable, that often changes this trade-off. You're more likely in a small study to really get a benefit out of this. You want to reduce the variance so badly you're willing to accept the bias, and your inferences are still better by doing this. So in a rare disease setting, for example, this also comes up, and the bias variance trade-off is more likely to be in your favor.
Kurt,
yeah.
Can I ask a question? The bias you're talking about here is more like bias in the rate, but like particularly in the rare disease setting, you're talking about Isn't there kind of an additional source of bias that could come into play in that you're really only doing the trial because you had results you liked in the smaller study? And is that being accounted for here, or is that a bias?
No, that part, that's a bias, yeah, so that, it's one of those interesting, there's two components to the bias, and so I gotta be careful, you're exactly right. This is sampling variability, statistical bias, and then there's scientific, which depends on what your definition of statistical is, but there's definitely two components of it, and it's not including that second piece.
So from phase 2 moving forward, I have a biased estimate of the effect. How would I move forward to the next level? If, I've reduced the variability, I've shown the benefit of the treatment, but.
Well, so it's an interesting question. So can I table that question until the end?
Sure.
So, because I have a food for thought at the end of this.
Okay.
So, you ask all the good questions, I refuse to answer all. So essentially, we're going to run out of time, and I'm going to flee the room. So let's see. So I talked about this. So this just highlights the high weights and the low weights. So let's do this. Here's what happens from a testing perspective, and it's important. Testing actually works differently than estimation. Estimation has this pretty broad sweet spot where things work well. If you test, the risk profile is a little bit different. So this is the type 1 error graph. And so you can see the orange one here, type 1 error is controlled a little bit below 2.5% across the board. If you borrow, if you're in a situation right in the middle, so you have a minimal amount of drift, the type 1 error is actually reduced. And one of the reasons is, if you think about how you can get a type 1 error, you can get it, so in truth there's no difference. You can get a type 1 error if you get a random high on the treatment arm. You can also get a random error you can get a type 1 error if you get a random low on the control arm. In effect, historical borrowing lowers the chance of getting a random low on the control arm because it pulls things in as long as you are in this region. So you do decrease the type 1 error. On the other hand, and certainly if you're on this side, so I have to be careful, on this side you get reduced type 1 error. That's because all the biases make it harder for you to win. And making it harder for you to win reduces the type 1 error. On the other hand, if you're on this side of the graph, then the type 1 error is going to be inflated, and it can be inflated a lot. So you've got to be really careful on how quickly these things. So typically from a regulatory standpoint, this is the most terrifying part of the graph. This is where you'll have all your discussion. And this makes sense. I want to show what the single arm trial does. So this is why single arm trials are so risky. If you see this purple, that type 1 error quickly goes off the graph and quickly goes up to 100%. So if you do a single arm trial and you encounter, hey, well, the controls just got better over time, you're never going to know because there's no test plan. You're just going to declare, hey, this works, when in fact just everybody's doing better now today than they were yesterday. The flip side of this is the power. Again, the same process is going on. If you're on that side, it's easier to win. If you're on that side, let me take that back. If you're on this side, it's harder to win. If you're on this side, it's easier to win. In terms of power, being easier to win is a good thing. So you can see that these power graphs, they're actually above the no borrowing orange line, but there does come a point where they start to go down. So if you've had drift in the opposite direction, you can lose power. For the most part, that doesn't happen very often, but one example is antibiotics, for example. People get, pathogens develop resistance over time. So it's conceivable if you're developing an antibiotic that you had a 65% cure rate sometime in the past, and now more patients are resistant, and now you're down to here. That can happen, and borrowing in that case potentially can reduce power. That's less likely in most practical settings. So usually you're more worried about, well, this type 1 error, and you get the gain in power out of this. So again, I just want to describe these kind of regions. If you have a minimal amount of drift, everything's in your favor. Mean square error, type 1 error, power, they're all up. If you go off to the sides, either way generates bias. If you go off to the side in this direction, you've got a type 1 error worry. If you go off in this direction, you've got a power worry. So it's important for you to consider in advance, where do I really think I'm going to be? Am I going to be in the situation where I get a gain? Or am I going to be in the situation, how much risk do I have that I'm going to have a problem? We often quantify this, so you can, for example, elicit a prior on where people think this parameter might go, and you can compute expected type 1 errors and expected powers out of this, and you can start to quantify that risk for people to make decisions. All right, so let's see. Okay, so one thing that you also should see here is that if I have this drift between them, so this difference makes such a big deal, if I knew it in advance, I could probably do a much better job of picking the weight. So if I knew I wasn't going to get any drift, well, I'd probably want to use a really high weight and get the benefit out of it. On the other hand, if I knew that my drift might be fairly large, maybe I want to pick a low weight, because remember that mean square error, I got a benefit over a really broad range. So, but of course you don't know it in advance. You do get information during the trial on what it is. So this gets back to your question on if at the end of the day you got into a situation where, boy, I got a pretty big discrepancy, I'm not very happy I weighted it so highly, can I weight it less? You can do that from the standpoint as long as you pre-specify the analysis that takes into account for that weight. Oh yeah, this I love this kind of quote here. So if you've ever seen this, those who cannot remember the past are condemned to repeat it, this is the exact opposite of what we're worried about. If historical borrowing, if we could just go, yes, we will repeat the past no matter what, everything would be fantastic. We're effectively worried about it. The history will not repeat.
You need to quote Heraclitus.
Which is that quote?
You can't step into the same river twice.
Ah, okay. That does make sense. All right, so dynamic borrowing. So this gets at the notion of, can you use the data to set the weight? I'm not going to go through all the details here. There is in the slides, it's in the paper. But in effect, what happens is you fit a hierarchical model. So this part is exactly as before. So I've got my current control data, control rate, historical data, historical rate. What I'm going to do is I'm going to put them into hierarchical model, and I'm going to fit something akin to a meta-analysis, pretty standard meta-analysis approach for the control arms. What this does is it explicitly has a parameter, and it's this tau parameter in here, that measures the across-study variability. And you can write, there's a paper that shows there's a one-to-one relationship between the value of tau and the weight that you put on the data. So, it's not an easy formula, but there is one, it does exist. So if I just picked a tau, then I'd be just picking a weight. I put a prior on tau, and so in effect what I'm letting the data do is I'm letting the data pick the weight. More precisely, the data is giving a distribution over the weight. What that does is if I see a big discrepancy, that posterior distribution will shift towards lower weights. If I see the two things are very close together to each other, so if let me make sure I said that right. If the current data and history are very far apart, tau becomes large and I weight small. If the historical and the current data sets are close together, then tau moves towards smaller values, and that corresponds to higher weights. So in effect, this is a way to do this dynamic reweighting, but I'm specifying it in advance.
So in effect, the prior on tau, the distribution of that prior parameters in there, they are estimated from your data, or?
You have to specify the prior, but the posterior comes from your data.
Okay. I was thinking more like if you said it non-informative, or some of the parameters are fine-tuned to go back to the data.
So we usually pick the parameters of that prior on tau to generate nice operating characteristics.
Okay.
So you don't have a lot. Tau is an across-study variability parameter. You don't have a lot of studies. You're never going to estimate it with any kind of incredible accuracy. So usually what we do in this case is we plot operating characteristics and pick one that does reasonably well for the context. So this is the behavior of this mechanism. So, something that, okay. So anyway, we talked about when you picked a weight and you weighted your data 20%. If I had 100 historical subjects, I weight them 20%. I'm effectively borrowing 20 observations. If I pool, I'm borrowing 100. This graph is giving you the expected number of borrowed subjects. I now have a prior on tau. So the weight itself depends on the data, so the weight itself is random. So I can compute its expected value. The different graphs here correspond to different priors, and this gets back to your question. Eventually you do have to pick one of these priors as opposed to the other. But you can see under all of these priors, what it does is the closer the true control proportion is to the historical data, you tend to get higher weights when they're close, and you tend to get lower weights when they're farther away. So there's this natural automated down weighting. So for example, this solid green one, which we're kind of fond of, this one has, near the middle, you actually get a weight that's closer to 0.4, closer to a half. But as you get this amount of drift, it starts to go down. So by the time you get out to 55%, your weight's down to 1/10. So it's naturally, and that's an expected value. That's not a guarantee, but in expectation, you tend to get small. These end up being negative, which is kind of an artifact of how we compute expected number of subjects in a posterior. For normal distributions, it's nice and flat, but for dichotomous, it's a little awkward. If you look at the mean square error, this is what the mean square error of this green curve looks like. So the cool thing is you get a lot of the benefit when you're in this sweet spot. There is an issue where it's inflated, but you have in effect bounded your risk. So this goes up a little bit, but as the discrepancy increases, it goes back down, and you can see this is going back down to no borrowing. This will get down to no borrowing again on that side. So again, if there's no drift, this works as well as using a weight of about 0.4 or so, but we've avoided some of the risks on the extremes. Same kind of situation for type 1 error. We again get bounded. It's inflated, no question it's inflated, but it's bounded, and the power has the same kind of, we've mitigated the power risk. So this is a notion of trying to keep a lot of the benefits while mitigating some of the risks.
Kurt, can I ask this question?
Yeah.
When you're dealing with like the regulatory authorities, the increased risk, are they okay with that given this? Or usually that's
Yes.
a big sticking point.
Well, so I should be careful. So it depends on where you are. CDRH, it's actually, as I said, it's explicitly in the guidance now that they're okay with it.
Okay.
You need to have a statement of what it is. So they want to see a type 1 error
Of
curve.
the bound.
Yeah, they want to see the bound and know what you've done. But depending on the context, they're quite comfortable with. I've recently had one where we did it one way and they actually suggested you can go a little higher, which was, that was a pediatric indication where they wanted to borrow heavily and they were happy with it. So it depends on context. They want to see it, but yes. So, Lissa Levange has talked publicly. She is okay with this in terms of doing this. Now that said, if you go in certain parts of the FDA, you will have, there is no such thing as the FDA. But anyway, yes, they are okay with this.
I have a question. I just have
Yeah.
a question.
Oh, please.
So just looking at this, why, and the unknowns and drift, why would one pursue a fixed weighting?
I would never recommend for doing a fixed weighting. So it is, I would a fixed, well, so I should be careful about that. In some small studies, there is not enough information to accurately estimate. There's so much variability, you don't estimate tau very well. So there are situations where they're somewhat close. I actually just got a request to review a paper on this, so I may tell you something different tomorrow.
And along those same lines, I've sometimes seen analyses where rather than pre-specifying a certain way, a range of weights is shown and how that would affect the results. Is that's more done, I guess, in sensitivity analysis?
Yeah, I think after the fact, you definitely can do sensitivities along those ways. This would be, if you're pre-specifying, you'd want to pick a weight in advance.
How many historical data sets do you need to accurately do the dynamic farming?
So we do it, I mean, obviously the more the better. There's no question the more the
I
better.
mean, can you do it
We
with
do,
just
we do it
one?
with one. So you've got, if you do it with one, then there's sensitivity to the prior. So the fewer studies you have, the more sensitivity there is to the prior. But we do it with just one. And I mean, certainly we are not, there's no claim that we're estimating the across study variability well. But what we're doing is we're mitigating that risk regardless. Obviously, that if you have 5 or 6, life is a lot better.
So in, like, I mean, you talked about this a lot in, like, drug development, they thinking in more like the academic trials, like what the students in the course would be doing. I would imagine, the drift is probably a bigger issue because there's usually more temporal, it's longer with the studies, they may be in separate populations, and you probably have fewer. So this would work, you would just have the complications in terms of defining the drift.
You would have the
distribution.
complications in that, and I would be inclined to potentially, if I was worried about a lot of drift, I would want to make sure that I had picked a prior. I probably don't want to heavily, I don't want my prior to be one of these where I'm still going to heavily weight, even though I get a lot of drift.
Okay.
So I think you have a discussion over what range do you think you're in. If you're overwhelmingly likely to be in this range, I'm going to act differently than, I could be anywhere.
And is there a point where, like, if you have, maybe you have lots of early data, but you know there's been a drift, right? I mean, like, you know the standard of care has changed. Would this still help you, or would this be too problematic?
You could try to model, put in a shift and do it, but I think you're starting to get into problematic areas, yeah. I think you've got to have a lot of faith in, you've got to jerry-rig this, and you have to have a lot of faith in that, so I would generally say no. Let me add one last thing. So here's my food for thought. And this gets into, should I really worry about that type 1 error? So here is.
So even if we have the drift, we are reducing the variability.
Oh, you're always reducing the variability. You're always reducing the variability regardless. And sometimes by a lot. So suppose I did this. I sit in R, I open it up, I just type in PC and PT, so I type in here are the rates. I don't show you what it is. Then what I do is I generate binomials from PC, and I get 6,399 out of 10,000, just a random draw. And now I tell you, design a study. You're going to be able to enroll 200 observations per arm, and I'm going to guarantee you that I am not going to make changes to PC and PT, and I'm a trustworthy guy. So I'm not going to change it. There is no drift here. I have now there's no systematic drift. There's sampling variability. You've still got sampling variability there, but there's no systematic drift. By design, I have forced that I have ignored all worries about this. What are you going to do? So I claim you have to pool this data. This is real data on the control arm. You know it just came from PC. There's no rational way that you're going to ignore 6,399 out of 10,000 in favor of the 200 in the current study when I just told you I used the same parameter. But that doesn't mean there's not still sampling variability. So I could still generate those type 1 error graphs, and I could still have drift due to the sampling error, and those type 1 error graphs, if I got a random low, then I'm going to get type 1 error inflation. But you're still on a pool. And I'm just going to leave that kind of as a bomb in the room. So anyway, that's usually the typical argument on why you would accept this. If you think that the data was on point enough, the type 1 error is more likely to be a result of sampling variability and not systematic drift. And that justifies taking the type 1 error, because over the long run, you will decrease type 1 error more often than you increase it. So from the FDA's long-run standpoint, they will make fewer type 1 errors. They're just not sure which studies are inflated and which studies are inflated.
So if you were designing a concurrently controlled study, knowing that you have access to historical controls from there, what you showed was the expected sample size that you borrowed. So when you design a concurrently controlled study, would you design it in the typical way, assuming that you're just comparing that and expect that those additional borrowed subjects are just going to give you benefit over what you designed for? Or are you going through this process of simulating the hierarchical model for the borrowing and incorporating that to come up with the sample size for the current study?
So we would typically, we would design the study, so we often, there's one example in here. So this is an antibiotic study, I know we're out of time. There we go. Anyway, so I can't find it. So we had an antibiotic study, typically a 750 patient, 375 per arm study. They had historical data that they were going to bring into this. What we did is we did change the sample size down to 600. The other thing we did is we switched it from a 1-to-1 to a 2-to-1 randomization because we're borrowing on the control arm. So it made sense to enroll fewer controls and increase the number of treatments. So what was cool about that is that actually you got 375 per arm. We decreased the sample size by 20%. It's 200, 400, and we actually have a larger safety database because we've got 400 patients on control rather than 200. So we would typically make some design adjustments to account for the fact that we're borrowing on controls and not treatment. So often these are accompanied with unequal randomization. But you absolutely could, I'm going to design it, this is what I was going to do anyway, and I'm just going to get an extra bit of benefit. The question is, how do you want to take that benefit?
Is there any way to account for in your model, like your hierarchical model you set up, where I would imagine a common problem, especially in the academic trial setting, is maybe you have preliminary data, but it's on a small number of sites, and now you have more, so you know that your drift could mostly be due to site-to-site variability. Is there a way to account for that in the model?
I would think yes. I haven't seen any work that actually explicitly does it.
Okay.
We've also, the thing that I have seen recently is mixture models instead of just that prior.
Mm-hmm.
There's a mixture model where you've got kind of a point mass on tau equals something really, really large, which corresponds to essentially a weight of 0. And that gives you more of an explicit off-ramp where you've got, hey, if the discrepancy gets big enough, I'm not going to borrow at all. And it's just more explicitly in the model. I kind of like those these days, and that's one of the things we've been working on.
Could you share some good papers or the link to the papers whenever you have time on some of these designed and published, which are published or?
Yeah, absolutely. If you email me, I've got, so that pharmaceutical statistics, that's a review paper, and it goes through, it has references to lots of other papers as well.
I'd be curious too, you mentioned a lot of the FDA discussions, but have you had any experience with how grant reviewers react to proposals of using this?
Not as much, no. What's kind of interesting, I kind of personal peeve, so the answer is no. It's always interesting when I don't want you to incorporate it in the study, but of course all the grant reviews go, well, we know from paper X and we know from paper Y and we know from paper Z. It's sort of like, you're doing it too, just not formally?
You're putting a point mass instead of really.
So yeah, I think intuitively we do this all the time. This is just quantifying what the risks are.
You'd mentioned at the beginning not cherry picking, subjects or even studies. I could imagine a case where historical data might, there might be clusters if you have enough historical trials. Do you always treat them all sort of equally?
Nope, we have fitted Dirichlet process mixture
Okay.
instead of the hierarchical model as well. So you can do that. It does nice things on the type 1 error. So we haven't published that yet.
Oh, come on.
Mary's pretty bad about publishing. Not our strong suit these days. Anybody wants to collaborate?
Can we get a copy of your slides?
Oh, well, whatever, just email me. I
think Sam will collect them. She
Sam
usually
has them, so I'm happy. I mean, I'm happy if Sam just emails the entire course.
Yeah, I think she usually sends an email out after to ask if anybody has problems with them being shared, and then I think she just sends out.
I will certainly have no problem.